Well, Dr. Clark here, and today we're going to demo out lab number 6, or Microscopy and the Cell. Okay, so the first thing in this lab, okay, is parts of the microscope. Okay, now parts of the microscope will depend a lot on the type of microscope, the brand of the microscope, and all the gadgets that might be added to the microscope. But real quick, I'm going to go through the 2 different types of microscopes that are most often used in Gen Bio classes. And then you can see by the diagrams that are on page 42 in your lab manual that shows you the pieces of the microscope. Okay, the microscopes that we use are they're sketched there with the pieces labeled on the next page. Just be familiar with those pieces. Now, as I progress through this lab, I'll talk more about different features like the eyepieces, the objective lenses, the stage holder, the iris diaphragm, these other features of the microscope, parts of the microscope, and what's the purpose and how to manipulate them and why you might want to change the iris diaphragm to allow more or less light to come into the microscope. Okay, so we're going to skip over that, and the first thing that we want to do, we're going to skip over the individual parts, but the first thing I want to do is introduce you to the 2 types of microscopes that we're going to use in this course. Okay, so the first one is this microscope here. This is called a compound light microscope. Now, some nice or interesting features with compound light microscopes is they normally have numerous objective lenses. Okay? And each objective lens has a different magnification. For example, the one in place right here is a 4x objective lens. Okay? And that's sometimes is called low power, other times people call it the scanning objective lens. But nonetheless, that's where you want to start the microscope when you start focusing the microscope. Okay? And I'll get to focusing the microscope here in a second. The next one is 10x. Okay, which is called medium power, or sometimes people call it low power. If they're calling the first one scanning, then they call the next one low power. The next one is 40x. Okay, that's almost always called high power. And the final one is 100x, okay, which is always called oil immersion. Okay? Oil immersion means just what it sounds. That lens needs to be embedded in oil. Okay? And so there's a special oil that we use, a microscope oil that we use, and you put it on the slide, and then you rotate the lens into the oil, and the lens gets coated with an oil, and this allows for a more condensed light beam to come through the image. Okay? So when you're looking at, 100x and 10x eyepiece, you're talking about 1,000 times your eyesight. When you're looking at something that's 1,000 times your eyesight, or very minute, you need a lot of light. So as we're progressing in the lab, you'll see me turn the light up as I move through the different objective lenses. And that's because with every magnification, or every increase in magnification, often you need a little bit more light to light that image in order to make it a visible image. Okay. The next piece of equipment is a dissecting microscope. Okay, you can see it's very different. The stage, or the base here, is open. Okay, there's nothing containing. And this is so you could actually put something on here and dissect underneath it, just like the name of the scope suggests. Okay. The other thing about dissecting microscopes is there's only a single lens. The lens is inside, and to adjust the lens, it's just a series of mirrors, but there's a adjustment knob here with some magnification values on it, like 4 here, if you can see that, is basically telling you that that's 4 times your eyesight. And you can turn it to 1 times your eyesight, 1.5, 2, 3, 4, 4.5, 4.5 times your eyesight. Okay. Now some dissecting microscopes will have eyepieces that have a magnification themselves. So if you look at this eyepiece here, it says 10x. So you do simple math. You take the eyepiece times whatever the magnification here. In this case, it's 4.5, so that's 45 times your eyesight. And over here, same deal. Okay, these eyepieces are also 10x, so you can see right here there's a 10x, and the one that's in place is a 4x. So these are very close, and if I just turn this one to 4, these magnify the exact same. This one's magnifying the object to 40 times your eyesight, and this one's magnifying the object to 40 times your eyesight. The dissecting microscope tops out at 45 times your eyesight. The compound light microscope that we have here, it tops out at 1,000 times your eyesight. Okay? So 2 basic microscopes. They have very different uses. Okay? The compound light microscope is normally used to examine things that are going to be on slides. Well, always going to be on slides. The dissecting microscope, you can use slides on it, but often it's used for things that are not going to be mounted to a slide, but rather can be placed in something like a petri dish or in a dissecting tray or something like that, and you can manipulate the organism while it's under the scope. Okay, so you can get to see different sides of the organism, or you can dissect it, or whatever you want. Okay? All right, so now I'm going to go through the lab 6, and I'm going to go through the different procedures. So we skipped procedure number 1, sort of, which is just identifying the parts of the microscope. I'll let you guys do that because it's fairly simple. You can do that on your own. Okay, procedure 2 is going to be filling out table 1 in your lab manual. Okay? And that means that we're interested in the orientation of E. Okay? We're interested in what kind of magnification does the scopes have. We're interested in the field of view, so how big or how much area can you see when you're looking through the scope. Okay? And then we're interested in the depth of field, so how deep can you look, okay, through an object. All right, so we're going to go at that in a couple different ways. First, the first part of that procedure is we're going to examine the letter E. So if you can see this, okay, you can see that I have a slide here. The letter E is on that slide, and I'm going to examine that under the microscope. Okay? And right now our microscopes are plugged into the computer. Okay? So you're going to see exactly what I see on the microscope. So I'm going to start with the compound light microscope. I'm going to start with the scanning objective, okay, or 4X. I start by placing the slide on the microscope. There you can see it on the microscope. And then using the slide adjustment knobs, I'm going to move the slide so it's in the center of where the light is shining through. Okay, and that's this piece right here. I don't know if you can see that, but this piece right here is the iris diaphragm. Okay, and it has a controller on it which you can open and allow more light to go in. So this is the diaphragm lever, or you can take light away. So you can either open it, allow light to come through, or close it, and you'll see that often, especially when later on when I start looking at pond water and things like that, or plant leaves, I'll adjust this to let more or less light to go through. So as we start, I'm going to leave it all the way open. That lets the majority of the light through. Again, I have the scanning objective lens in place, and I want to start always with the stage as far up as it can go. Okay, so using the coarse adjustment knob, that's this knob here, I'm going to twist that until it doesn't go any further up. Okay, and then I know, okay, that's as far as it goes up, and then I know I only have one direction to go to focus this image, and that would be down. Okay, and so I can do that by just using the coarse adjustment knob and rotating it towards me. I can start moving that object down. Okay, now because this microscope is on scanning, you can see that I already can see the letter E. Now, it's not quite perfect, so it's not really crisp, but nonetheless, you can still see that the letter E is there. Okay, so I can adjust the coarse adjustment knob, and I can crisp up that letter E a little bit, and you can see there that that letter E is now fairly crisp and clean. Okay, and you can see from the original letter E that I showed you, okay, and I can show you it again on another slide if you can see that. The letter E that was on the original slide, okay, was upside down and backwards. Okay? And you can see that the compound light microscope does that to the image. It flips it right side up, or right side up, and then the correct way, the correct orientation. So what that means is, using a compound light microscope, because of the system of lenses, it always flips images upside down and backwards. Okay? And that's important to know when you're trying to find something, like maybe you're trying to find, maybe you're looking at a root of a plant, and you need to know where the root cap is, and that would be the tip of the plant's root. Well, if you throw it on there and you start moving, okay, you actually need to move in the opposite direction that you think it is. Okay, and then because that image is going to be flipped upside down and backwards, and then you would say, well, that's not technically the right hand side that you're looking at, it's actually the left hand side. Okay, so and you'll understand as you progress, or after you use a microscope a few times. Okay, so I'm going to come over, do the same thing on the dissecting microscope. So I put a slide on the dissecting microscope. It's got a letter E, just like the other one. Okay. Now with the dissecting microscope, this is a matter of, it really doesn't make a difference where you start on this. A lot of times you're just going to have the head of this microscope far enough away from your object in order for you to get the object underneath. So if it's an insect or a plant or something like that, you don't want it right on top of it, crushing it. So you want to give it some space, and so I have plenty of space here. I have it at 4 times 10, so 40 times my eyesight. And then just like the other one, except for instead of moving the stage on a dissecting scope, I'm going to move the head of the dissecting microscope using this coarse adjustment knob. Same thing. So now I'm just going to move it down until I can see the image or see the letter. And because my camera, this camera is kind of out of whack. Okay, so sorry about that. Camera's not tight. As you can see here, there's the image. I can manipulate that material, so that's one of the nice things about a dissecting microscope, is you can manipulate the material so you can get a better image of it. And you can see what this scope is doing to the image is it's keeping it upside down, but it reverses it. So it is flipping it around. Okay, so different microscopes manipulate material in different ways. Okay, so this is just upside down now. Okay, on the slide it's upside down and backwards. Okay. In the case of the compound light microscope, on the slide it's upside down and backwards, but compound light microscope manipulates it so it's right side up in the correct orientation. Okay. All right, so we'll move into the next procedure in just a second. Hi all, I'm back for the next procedures in the microscope lab. Okay. And so this next procedure is to examine the magnification or to list the magnification that each microscope has. Very simple, okay, a simple calculation. In the case of the compound microscope, you just take whatever your eyepiece is, okay, or your ocular lenses are. These are 10x, and you times it by whatever objective lens you have in place. So your scanning objective is 40, okay, and then 10x times 10 is 100, 40 times 10 is 400, and then oil, which is 100 times 10 is 1,000. Okay, so that is the magnification that this microscope is capable of. Now that can be adjusted. You don't necessarily have to get a new microscope. You can change these objective lenses. So maybe you have a 2.5 times objective lens, or maybe you have a 15 times objective lens. You could also change the eyepieces. Instead of having 10x eyepieces, you could do 2.5 or 5x eyepieces. So there is ways to manipulate the magnification in a compound light microscope. Okay, we jump over to the dissecting microscope. Okay. And again, we discussed this before. It has a 10x eyepiece, okay, or ocular lens magnification, and then it has an adjustment knob here. Now the difference between the two is when I am looking at this, I can manipulate this by steps. So we can start all the way down here at 0.7, okay, times 10, so this is 7 times your eyesight. But the next one is 0.8, or 8 times your eyesight. So if I go halfway in between the two, it's still a magnification. So now this would be 7.5 times your eyesight. And if I do about a half turn, or a little halfway in between the next two numbers, so it's 0.8 and 1, so halfway in between that would be 9 times your eyesight. You can go to 1, 10 times your eyesight, etc. So the dissecting microscope allows for you to manipulate it at smaller increments, okay, but clearly you don't have the magnification that you can get to on that microscope. You can't go all the way up to 1,000 times your eyesight. Okay, well, you wouldn't want it anyways, because you're not going to dissect the bacteria underneath the microscope. Okay. All right, so moving from there into the field of view. So now we're interested in how big or how wide of an area can we see under the different magnifications. So you can see here I have a ruler that's placed on the microscope, and on the metric side of the ruler I'm going to find out how many millimeters, okay, I can see while looking underneath the microscope. Now, it's deceiving when I show it to you on the computer screen. You can see there's the ruler. These are the tick marks on the ruler. Okay, a millimeter is the distance between the tick marks, the inside of the tick marks. Okay. Now, the reason why you're going to get a different number if you're looking through the microscope than if you're looking on the computer screen is the field of view between the two is very different. You can see where the microscope ocular lens come in versus where the microscope's camera comes in. Okay? That changes the field of view. So the magnification is going to be changed, so getting the camera to be in focus, it's not going to be in exact focus the same time the eyepieces are, but also the amount of, or the field of view is going to differ between the two. So if you're truly interested in the field of view that this microscope gives you, you can't do it by examining the computer screen. You have to do it by looking through the eyepiece. So if I look through the eyepiece, I can tell you that the field of view of this is 1, 2, 3, 4, 4.5, or close to 5. Here on the computer screen, you can see 1, 2, maybe almost 3 millimeters. So it's very different. Okay, same thing with the dissecting microscope. Okay, if we come over to, again, 40 times my eyesight, right, and I look through here, 40 times my eyesight is 1, 2, 3, 4, 4.5, close to 5. We look at it on the computer screen, and you can see there that it's 3 with a little bit, maybe 3 1/10 or 3 2/10 millimeters. Okay, so from that standpoint, you can see there's a very difference between looking through the eyepieces or the ocular lenses and classifying the field of view and looking through the camera. And you can see again the distance between these two are different, the size of the tube is different, so that gives you a different field of view. Okay? But nonetheless, that's how you calculate the field of view, is by examining the number of millimeters between each, or with each magnification. So for the dissecting microscope, I'm going to real quick do the numbers. Okay, I'm going to start at the lowest magnification, which is 0.7. I'm going to count the number of millimeters. So here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 millimeters or so. Okay. And then I'm going to crank it all the way up, and that's according to the computer screen. I'm going to crank it all the way up. This is 4.5, or 45 times your eyesight. According to the computer screen, we have 3. Okay. That's the easiest way to show you the data, so I'll just use the computer screen to show you the data because you won't be able to see it through the eyepiece. So here is the lowest. This is 40 times your eyesight. Again, this is roughly 3, almost 3. Okay, I'm going to switch, move up the magnification to 10 times your eyesight. Now I'm going to move this just a little bit so I can have a line in the middle, and you can see, oh, actually I'm going to move it so I have a line on one edge. So you can see that this is a little over 1 according to the computer screen. Okay, so 1 millimeter, and then roughly whatever this is, so maybe 1.5 millimeters according to the computer screen. I'm going to move it up to 400 times your eyesight, and now it will become very difficult probably for you to determine, because there you might only see black. So this is only black here. I'm going to increase the light. So you got a little bit of white over here and an entire black line, and maybe a little bit of white over here. So under 400 times your eyesight, your field of view is less than 1 millimeter. Okay? And it's not possible to do oil immersion. Just know that it's going to be even less than that under oil immersion. Okay? We'll come back and we'll do the depth of field. I'm back, and so now I'm going to look at the depth of field for each of the microscopes. Okay? In order to do that, I have a slide with colored string or colored thread embedded on that slide. Okay? So what you're going to see here on the computer screen is you can see that there are 3 strings. And what you should see is at 40 times your eyesight, or with 4x objective lens in place, you should be able to tell that the blue string or the purple string is on top of the red string, which is on top of the yellow string. So I think most of you will be able to see that. Now, we're interested in what happens to the depth of the field as we increase the magnification. So now I'm going to go to 100 times your eyesight. Right, and I'm going to try to focus on the top string. But now you can see the blue string. Okay? You can probably still, it probably still looks like the blue string's on top because it's in focus. But now the red string and the yellow string are not in focus. So now you already know the order in which they appear, okay? But because they're not in focus, it's very difficult to tell which one of these strings is on top of the other. Okay? And so as we increase magnification, the depth of field decreases. So this is important because as we increase magnification, you might be looking at the top layer of a plant leaf or the top layer of a cell, and then you increase the magnification, and now you could be looking through the top layer and into the middle or to the bottom layer of the cell. Okay? And that's important because different things occupy different spaces on objects that you're interested in. So for example, if we're looking, let's say we're looking at a plant leaf, and we're looking at the top layer of the plant leaf. Okay, that's where the epicutical layer is, and then we're interested in magnifying that epicutical layer, but instead we look through the epicutical layer and we're looking at the bottom of the plant leaf now. Well, that's where the stomata or the pores are. Okay, and so you might think, okay, well, there's pores on the top of the leaf, and majority of leaves do not have stomata on the top of the leaf. So it's important to know where you're at, what plane are you looking at when you're examining an object. Okay, so that's 10 times your eyesight, or 100 times your eyesight. Here's 400 times your eyesight. I'll have to increase the light that's coming through. Let me see if I can get some more light to come through, and you're going to see that it's going to be really difficult for you to tell which one's on top. I don't even know if I can get it to focus on the computer screen to see which one's on top. So I'll have to move it out from the center here. And there you can see the blue thread. It's in focus, but when I have it in the center, it just looks like a big black spot. You can't focus on anything. You can't focus on the top or the middle or the bottom. You have to move it to the outer edge. And you'll see that that's important to know also, that if the thing that you're trying to look under the microscope is thick, then you won't be able to focus at all. So you'll have to go to the outer edges where it's much thinner to get a focus point. Okay? We jump over to the dissecting microscope, and I have the three threads here. Okay. Let me see if I can turn the light down just a little bit and get those to come into focus. All right, and we're going to start at the very base there. Okay, and I think most of you can see here that the red line is on top, and then it goes blue and then yellow. Okay, and as we increase magnification, that is not lost. Okay, you can still see. Let me make sure that I'm getting it to stay in focus as I change the magnification. But you can still see all three lines. They're still, oops, sorry about that. They're still in focus. I'm going to turn the light down a little bit. Okay, all the way to the same point that we were on the other microscope, 40 times your eyesight. And just like the other microscope, at 40 times your eyesight, you can see all three colors. You can tell what the pattern is. You can focus all three. Yellow's in focus, blue's in focus, red's in focus. Okay, and so from a dissecting point of view, the dissecting microscope, the depth of field does not change with magnification. At least not our dissecting microscope. So going from 7 times your eyesight to 45 times your eyesight doesn't change what you see. Okay? It doesn't change what's in focus. On the other case, when we're talking about the compound light microscope, as you increase magnification, you decrease depth of field. Okay? We'll come back and we'll start looking at some other slides. Hi all, I'm back. And now I'm going to show you the method to create wet mounts or create slides from different types of material. Okay? So in this lab, you're going to examine things like pond water, and you're going to see what creatures are floating around or swimming around in pond water. Okay? You're going to make a wet mount of plant tissue, so you can see chloroplasts and nuclei and vacuoles, cell walls, cell membranes, cytoplasm, etc. You're going to make a wet mount of yourself, your cheek cells. Okay, so that's an animal cell that we're going to examine. Okay, and then you're going to examine some other slides that are pre-made. Some bacteria slides, which have been pre-made, and then you're going to examine an insect, okay, which has been pinned to a board. So first I want to show you the method to creating a wet mount. Okay, so what I have here is an onion, and the nice thing about this onion is growing, you know, some new vegetative material. So this is the tuber, this is underground, okay, and this is the vegetative material that's coming up. Well, the reason why I say that's nice is we're interested in both the green part of the onion, or the yellowish green part of the onion. That's the part that's going to do photosynthesis and be above ground. But we're also interested in the white part of the onion, and that's the part that's going to store energy. Okay? It's going to be manipulating, it's going to be growing, it's going to be, the nucleus is going to be dividing, and the cells are going to be growing and multiplying. So what we want to do first is grab a clean slide, okay, and place a drop of water on it. Now, the reason why I'm going to put a drop of water on this is because this is a wet mount of solid tissue or solid material. Okay, so I'm going to put a single drop of water in the center of the slide, okay, and then I'm going to take, and in this case I'm just going to take a scalpel, and I want to cut just a really thin, as kind of thin as I can get, chunk of material. I'm going to take a pair of forceps, remove that material, place it in the water, okay, and then I'm going to take a glass cover slip. Okay, this is to protect the scope so nothing gets on the objective lens of the scope, but it's also to hold the material down. A lot of times it'll allow the material to lay flat. Okay, and then I'm going to place that cover slip on that material. Okay, in this case I'm just going to provide just a little bit of pressure to kind of flatten that material out, and then you've made a wet mount. And so you have a little piece of tissue on there in water, okay, and now you can examine it underneath both microscopes. It's not necessarily needed to look at it under a dissecting microscope, but by making a thin cut, now under a dissecting microscope you might be able to see the outlines of cells at 45 times your eyesight. Well, no might about it, you can. You just can't go any further than that. So you're not going to be able to see the nucleus of the cells, you're not going to be able to see chloroplasts or vacuoles or anything like that. Okay, so there's a plant. Okay, you can do the same thing. Okay, but now because I'm going to move to pond water, now it's not necessary for me to place a drop of water on there, because pond water already is you know, aquatic material. So I'm going to suck up a little bit of material from the bottom, and then I'm going to place a drop on the slide. And pond water is very easy to make a wet mount of, so water is in general, and then you just place a cover slip on it, and there's your wet mount of pond water. Okay. The final one is a wet mount of myself. Okay. So I'm going to examine my cheek cells, and so I'm going to just take a toothpick. Okay. I'm going to take a clean slide. And because cells are not liquid, they're solid state, I'm going to put a single drop of water on the slide. I'm going to scrub the inside of my cheek. And then I'm going to smear that goo, that cheek goo, which is really just damaged cells. I just scuffed off a bunch of cells off the side of my cheek. I'm going to smear that in the water on the slide. Again, I'm going to add a cover slip. But this is not done, because your cheek cells, your skin cells, and everything like that, majority of the cells are actually completely clear. So in order for you to see the nucleus, potentially mitochondria, potential ribosomes, the cell membrane, where the cytoplasm interacts with the cell membrane, for you to be able to see any of those, you're going to need to dye the slide. But you can see how I did this. So cheek goo, water, cover slip, not dye. Now I'm going to add the dye. So I'm going to use methane blue. Okay, and I just need a small drop of methane blue on the outside of the cover slip. So I'm just going to put it on the outside of the cover slip, a drop of methane blue, and then naturally, because there's water on that slide, that blue is moving through. Now sometimes it might be necessary for you to take a paper towel or a chem wipe or something and put it on the other side of the slide and do what we call wick. So you're wicking that material through the slide. So you're going to wick it across the slide and dye the cells evenly as that dye moves across the slide. This is the best way to dye material on a slide, because then you make sure that it's uniform, it's spread across the water, it's dyed the slides or the cells evenly, and you don't have, you know, a bunch of big spots of blue in one area of the slide and none on the other area of the slide. Okay? And you may have to do this with onion. If you don't have the green part of the onion and you're just using the white, the inside part of the onion, you should add methane blue to that, because onion itself, the cells are clear, and so you got to add a little bit of dye so you can differentiate between organelles, cell walls, etc., cell membranes. Okay, that's the gist of how to make a wet mount. Now we're going to examine these different features on the microscopes. Hi all, I'm back. Now I want to show you the pond water. Okay, so I have the pond water sample on the microscope. Okay. And the key to examining pond water are things that are in liquids, especially if you're examining it for very minute, very small creatures. The key to it is get a little bit of sediment with it, or get something that you can focus on. So if you get a little bit of sand, decaying plant matter, decaying animal matter, or something like that, then you have a focusing point. So get one that's stuck. You can see one here that's kind of moving around. It was stuck, but let me see if I can find another one that isn't moving as quick. And once they get stuck, then you can get a better look at them and kind of analyze them and identify them. So here's one up here, right there. Okay, I'm going to focus in on this guy so you get an idea what that is. So that's another paramecium. So when I focus in, you can actually see the cilia, the little teeny hairs moving along the side of the body of this organism. Okay, and that's a good way to distinguish paramecium from something like euglena. Euglena will also have kind of a green tinge to it, but also Chilomonas will have two flagellums. They won't have cilia. These guys have cilia, paramecium do. And so I'll see, you can see some other things moving up here, some long structures moving up here. Sometimes we get like nematodes and other things, oligochaetes, so worms that occur in these freshwater ponds, pond samples and things like that. So move around and see if I can see something else. You also see really small things moving through occasionally, extremely fast, small things. Those are normally ciliates. Okay. They could be rotifers also, things that move fast, like here, that is a Chilomonas. That was moving with two flagella. Sorry, they're not exactly slow, so there's another paramecium. Okay, kind of trapped right there. And if I zoom in, you can see the cilia on that organism. Okay, and I'll come back after I find some more. I'm looking for an amoeba. There goes, you can just see some things shooting through the screen. That happens quite often. You just got to capture them. But I'll bring it back when I find something that's different than paramecium. Okay, so I'm back. As you can see, I actually brought you back to show you more paramecium. There are numerous paramecium in here. They're all over the place. I don't know if they're feeding on this detritus, this dead material, but you can see them throughout this water sample. There are just quite a few paramecium all throughout this sample. And some are moving, some are kind of trapped in the sample. And I'm still looking for some diversity. So like there you can see a paranemia. Oh man, so I'm kind of chasing it down. They're really large, and you see that flagella on it. There, it's kind of moving around, and see that flagellum. Okay, there's a different example of diversity here in this pond. Occasionally you're going to see some things that are hanging off of algae, stenators or stentors, or different species of stentors, and sometimes you'll see them moving, other times they'll just be hanging there. Okay, so that's it's a typical pond sample. And to examine these, you can see some paramecium, here, quite a few sitting there, some buzzing through, some stuck in, the algae. There's quite a bit of diversity here. Mainly paramecium, but again, quite a few varieties of things. I just going around looking at it. Lots of very minute rotifers, which are difficult to identify because they're really small. But got some dead paramecium. That's what are here and things like that. They're not moving. Sometimes it's nice to be able to look at those so you can see the cilia. So if I, really focus in on this guy, actually it's not dead. You can actually see it's consuming. It was eating things, and now it's moving, and now it's gone. Sometimes if you find one that is stationary, and maybe they're dead, maybe they're not, you can get an example of the cilia. You can watch the cilia beat along the sides. You can see these little rotifers and things flying through. Here you can see the feeding groove of this organism consuming material, and you can see it on the inside, and then you can see these little teeny hairs all along the outside beating extremely fast. Those are cilia, and so they'll have cilia in their feeding groove also, which is pushing food into kind of their mouth, I guess you could say, and that organism is feeding right there. So kind of neat stuff. If you've never examined pond water, there are a lot of neat creatures out there that are fairly unique and can show you a lot of life histories that are very different than, say, land organisms. So here you can see a stenotor. You can see it grow, kind of stretching out, okay, and I don't know if it's feeding. I'm not entirely sure what it's doing, but it's kind of got this loop. A lot of times they're filter feeders, so they'll stick straight up off of vegetation and consume material, but they have the capability of moving the trunk of the body, and there you can see that kind of movement there from that stenotor. Okay, so we're going to move from pond, and we're going to start examining some other things like bacteria, plant, and animal. Okay, I'm back, and we are now going to examine something under the dissecting microscope. And so the instructor choice for the dissecting microscope will be clearly up to your instructor, and I've just chosen to focus on an insect for the purpose of this demo. So if you're doing this online, then you can sketch the head of this insect. So you can see that I have, a swallowtail butterfly underneath the scope here. And I'm just interested right now on the head of this organism. And so you can see there on the screen that these are the eyes. This is a proboscis. The proboscis will curl when the organism dies, and then this organism actually only has a single antenna because the other ones broke off, but you can see an antenna that comes up here. This is a pen that goes through the body of the organism, dependent on there, but this is the the head of the organism. We have the capability of zooming in on that a little bit more on the eyes or the head of the organism, and so you can get a little closer, and you can start to really see individual pollen grains. And if we get even closer, you'll start to see the facets of the eye. So you can actually see the individual lenses of the eye there. Okay? And it gets, it kind of gets difficult to focus in on this once you get too far. It'll start to get a little bit difficult, but here you'll start to see the individual eye facets there. And so when people say that insects are their eyes have multifaceted, you can see all these little teeny dots. These are all individual lenses, okay? And insects have very unique eyes in which they see through each lens, and then they merge all the images, so behind them, to the side and front, etc., above them, below them, they merge them all into one image that goes into the brain, and both eyes will do that. So really, insects have remarkable vision in the sense that they can see pretty much 360 degrees. They can see above them, below them, behind them, in front of them, and that allows them to avoid predators, but it also allows them to find food and fly in different, habitats and things like that, and avoid collisions, etc. So can keep on zooming a little bit more, and there you can definitely start to see individual pollen grains that are stuck to the eye or even the hair follicles of the organism. The proboscis there, and whatnot. Okay, so that's that's really what a dissecting microscope is used for, is to examine larger organisms and get an idea of maybe morphology or physical features of that organism, and so you can get an idea of if we stretch this proboscis out, we can measure it, we can see how long the proboscis is, and that might be unique to this type of swallowtail. Lots of different reasons why you might want to throw something under a dissecting microscope. So here I have what's sometimes called a June bug or a June beetle, and they have a unique kind of head to them, and underneath the scope, that's what I have pictured here. And so here you can see the eyes, and then you can see these scoops, and those scoops are their antennae. So they have a very unique antennae. You can see the pedipalps, you can see these like feelers, and then you can see the mandibles, which are the pinchers or the biting mouth parts of this insect. Okay, so that's at 7 times your eyesight. We can go ahead and zoom in a little bit more, maybe just into a single eye, because we're starting to get or maybe both eyes. Okay, so there you can see the eyes. The eyes of these guys are marbled, or they kind of have a different coloration to them. Okay, so I can zoom in a little more. You can see all the hair-like follicles, okay, which are called trichomes. Let me move this up so I can get a good picture of that eye. I'm going to go ahead and turn the light down a little bit, and then focus this up so you guys can check out that eye. And you can see that they're multifaceted eyes, and they have a bunch of little teeny lenses. And you can't see that yet. I can get closer, and you can start to see the individual lenses of this organism's eyes. Okay, so just like the butterfly, this organism would take all those images that are created by each lens, and it would merge it all into a single image. And they have extremely good color vision, very similar to the butterfly. And you can see that they have unique trichomes in which they have some that are paddle shaped, and then they have some that are long, like hair shaped trichomes, and then they have hair or trichomes that occur along their antennae. Very unique organisms, very unique shapes, etc. Okay, all right, so that gives you another organism to sketch. If you're taking this online, there's another head of an insect to sketch underneath the dissecting scope. Well, I'm back, and now we're going to examine some bacteria slides, okay? And so, or bacteria samples. There's actually 3 samples, probably the most common bacteria genera on this slide, and that would be Cocci or Coccus, and those are circular bacteria. Bacillus or Bacillium, and those are rod-shaped bacteria. and spiral or spirillum, which are the spiral-shaped bacteria. So we're going to check those out. Now, the interesting thing about bacteria, or maybe not interesting, the difficult thing about bacteria are they are extremely minute organisms. So you need to go all the way to 1,000 times your eyesight, and then even at that, they can be fairly difficult to see. So here you can see splotches. Those are huge amounts of bacteria. So I'm going to continuously zoom in until I get to a place where I can go to oil immersion, and then I'm going to go to oil immersion, and you're going to see this. So here, even at 400 times your eyesight, you can just barely begin to see that these are rods. Okay, so I'm going to jump over to oil, 1,000 times your eyesight, okay, and we'll see what we get here. Okay, I'm going to turn the light up quite a bit and change the focus. Okay, and you can, oops, sorry about that. And now you can see that these are colonies of bacteria, and these are chains of bacteria, but the individual bacteria are rod-shaped. Now the color's not coming through very well, but these are purple. Okay, and for things that are purple under a gram-positive or gram-negative stain, that would mean that they're gram-positive. Okay? And we'll talk more about this when we talk about the bacteria lab. But these bacteria right here are bacillus, or have a shape of bacillium, so they belong to that genera of bacillus, and they're gram-positive, and they occur in chains of bacteria. Okay, so now I'm just going to slide over until I can find the next bacteria type. Okay, so here's another bacteria type. Again, under 1,000 times your eyesight. And you can see that these are not rod-shaped. And so these are, they can be linked together, but these are more of a corkscrew shape. So some of them make more of a corkscrew. A lot of these, you can see this one's kind of corkscrew, that one's a corkscrew. This is spirillum. Okay, and again, the computer screen's not doing it justice, but these guys are pink. So that, instead of being gram-positive, these are gram-negative. Okay. So spirillum, okay, that's the genera these belong to. Now I'm going to jump back and see if I can find the last. There we go. Okay, so the last type is the type Cosi, and you can see they occur in these large clumps, but if you look closely, you can see that they're actually balls of bacteria, or they belong to the genera Cosi or Coccus, and so they occur in these balls of bacteria. So if you have strep throat or streptococcus, a screen would show that in the back of your throat you would have these little teeny balls, and they would be gram-negative in the back of your throat. Okay, so that's bacteria, and these ones are purple. These are gram-positive. Okay, but again, it's hard to tell on this computer screen. Okay. Now we're going to move to another subject, and we're going to look at plant material. Okay, so I'm back, and now I'm going to throw on a plant sample. So this is that onion that we made a wet mount of, and I'm going to show you the onion in just a second. Once I get it on there and focused, I'm going to give you an idea of what the onion looks like under the scope. Okay, so I got it focused in. There you can see, try to get the glare off a little bit, and there you can see the onion. You can see these individual cells, so these individual rectangles. Some of them look circular, but a lot of that has to do with water being on the slide, and you're actually looking through a water bubble onto the slide. And you'll see what I'm talking about when I get to the edge and I show you the actual rectangular shape of plant cells. Okay, that rectangular shape or that rectangular line is the plant cell wall, and I'll point it out in a second. Okay, but again, this is what Robert Hooke was looking at when he described, well, not onion, but he was looking at cork, which is just the inside layer of bark, when he described cells to begin with. So cells were described via the rectangular shape that they possess, if we're talking about plants. Okay, so I'm going to move to 100 times your eyesight. Okay. And there you can see all these dark spots. Okay, and what those dark spots are is the stomata, or the openings of the leaves. Okay, so like for example, if you look here, you can kind of see that there's a circular or kind of jelly bean shaped on one side and another jelly bean shape on the other side. That's a stomata. Those jelly bean looking things, those are called guard cells. This is what opens and closes to allow for oxygen to leave the plant leaf and for carbon dioxide to enter the plant leaf. Okay, so we'll get another example, or we'll look at these in a little more detail. So that's the bottom surface of the cell. Okay, that's what we're looking at. Okay, now I'm going to go through the stomata and not be focused on the stomata, and rather focus on the cells that are underneath the stomata. And so you can start to see some of the rectangles that are occurring there. Okay, but we're still only 100 times, oops, sorry about that. We're still only 100 times your eyesight, so we're going to go further to 400 times your eyesight. I'm going to turn up, and I'm going to move away from the center. So the problem with examining the center of this slide is that you have numerous cells stacked upon each other, okay, because it's difficult to make a thin section through the cell itself. So I'm going to move to an edge of the cell and show you kind of some of those rectangular cells. There you can see the stomata again with the guard cells. Here, I'll show you those real quick. See these guard cells and the stomata opening? And now you can look into them. So I'm going through them, and so this is the outside. This is what you would see on the outside, and then this is kind of what you would see on the inside. You don't get to see those. Here's a good example. It's kind of difficult to see these, but these are rectangles. Again, these are a bunch of cells stacked on top of each other. But here's a cell right here, and you can see a nucleus in it, and there's another nucleus and another. And so there's quite a few nuclei here in all these cells. You know, by the looks of it, each one has its own. Okay, now if we move back into the region where, just a little bit over in the region, so here you can see these rectangular shaped cells. These are the region that's closer to where it's kind of colored or green, and if I change the magnification, you can start to see some like circular structures inside these cells. Those are chloroplasts, and they're the site of photosynthesis. So these guys are going to be yellow or green. In the case of this plant, it's yellow because it hasn't had a lot of sunlight. So that means most of the chlorophyll has been degraded, and just the xenophils, the yellow pigments, are left. But nonetheless, that yellow pigment is going to be inside all those little teeny circular chloroplast molecules. Let me slide around and see if I can show you another good example of these cells. Okay, so there I think you can, well, see here you can see these guard cells. These are the stomata. I'm looking through the bottom. But here you can see these chloroplasts, these circular organelles, pretty good. You can also start to see some things that are inside the cell that do not look like the nucleus. So here's a nucleus, but these don't look like a nucleus. That could be the central vacuole with different things inside of it. Sometimes they'll store enzymes and other things in packets in vacuoles. Okay, now I'm backing out. Come back in. See the nucleus of the cell. There's a nucleus. So you can see quite a bit. And some of you might be saying, well, I see things moving. You do see things moving. Okay, this plant was alive when I took the tissue. And what you're going to see is the cytoplasm, okay, the material within inside the cell, is flowing, and so it's called cytoplasmic streaming. And so organelles and other things are moving throughout the cell, okay, and they'll move constantly throughout the cell, so they don't become stagnant and just stay in one place. So a lot of what you're seeing is the guard cells. You're seeing the cytoplasmic streaming inside the guard cells of these stomata. That's what's showing up the best. But you can see it inside some of the other large tissue cells when I get in here. Maybe not in this region, but okay, so that's the plant. Pretty easy. You can see they have rigid, straight cell walls, and then on the inside of those cell walls they actually have a cell membrane. Inside that cell membrane they have cytoplasm. That cytoplasm is kind of like a jelly-like substance, and they'll have chloroplasts and other organelles. I mean, they have all their organelles, Golgi apparatus, everything inside of there. The nucleus is really showing up well in a lot of these cells. There you can see the nucleolus, okay, the region where ribosomes are made. So you can see quite a bit in these plant cells. Okay, so next we're going to move to animal cells. Okay, I'm back, and the last thing I want to show you is the animal cells, or the human cheek cells. So here you can see that I have the cheek cells under 40 times your eyesight or scanning objective lens. Okay, and it's kind of difficult to tell that there's cheek cells there, but we're going to zoom in and you'll see what they look like here shortly. So there you can start to see some individual cheek cells. Okay, that's 100 times your eyesight. Okay, I'm going to go 400 times your eyesight. Now you can really start to see the nuclei. Oh, I'm sorry. You can start to see the nuclei in those cells, and so you can see these cell membranes. The cell membranes are not rigid because these are animal cells, and you can see nuclei inside those. So lots of cheek cells. I did a really good job at scraping my cheek. Let me see if I can, okay, there's one. Whoops. Here's one kind of all off by itself. Okay, I'm going to go ahead and go to oil immersion and see if we can see some defining or other organelles with inside that cheek cell. Sometimes you can see ribosomes, or what seems to be ribosomes. Other times you can see a lot of bacteria because your mouth is full of bacteria. That's what I'm seeing right now, is just a ton of bacteria. On top of that, you can start see, if you notice that right on top of this cell, there's a bunch of bacteria. All looks like bacillus. Okay, rods. Now, whether it's gram-positive or gram-negative, no one really knows, or not yet at least. I could look at that, but I'm not going to. And I don't really see too much. You can see the nucleus pretty well. You can see the cell membrane. This might be two cells stuck together. That might be another nucleus there. Cell membrane, cytoplasm, nucleus, nuclear envelope. And I don't see the nucleolus on this one, but I'm gonna, I'll move real quick. Oh, here's one that's got a nucleolus that you can see pretty readily. So this cell right here, you can see that dark spot. That's a nucleolus. That's the site where ribosomes are made. Okay, and that is human cheek cells. So you can get an idea of what that looks like. Pretty simple procedure, easy to look at your own cells. Nothing too riveting, but also it's pretty cool if you've never done it. Alright, so with that being said, that should be enough for you to complete the lab if you're doing it virtually. Otherwise, this is enough to give you kind of an idea of what this lab's all about. Okay, till next time.